Hello, today I will be talking about Chapter 15, Chemical Control of the Brain and Behavior. Please make sure that you are practicing labeling a sagittal section of the brain as you are going to need to do this for your next exam. A great way to practice this will be utilizing the neuroanatomy worksheet that I created for y'all and then is posted on Canvas. Here's an outline of today's lecture. Chapter 15 largely talks about the hypothalamus and its role in homeostasis and the endocrine system. The endocrine system is this large system, including the hypothalamus, the pituitary gland, and other organs in the body that are responsible for either commanding or secreting all of the hormones that our body creates. There are three zones of the hypothalamus we're going to talk about. And we're going to discuss different types of cells that live in these zones, known as neurosecretory cells, that are responsible for commanding the secretion of different neurohormones and hormones. We're going to discuss the hypothalamus and its role in the autonomic nervous system response. If you remember, that is our involuntary control. We're also going to talk about diffuse modulatory neurotransmitter systems. These are essentially the areas of the brain where there are pockets of nuclei for certain types of neurons, such as serotonergic neurons or cholinergic neurons. I'll briefly overview the enteric nervous system, which is the mini brain in our gut. We'll discuss the pituitary gland and its function in secreting different hormones. The HPA axis, which stands for hypothalamic pituitary adrenal axis. And we're going to talk about how this is activated during the stress response to create a hormone called cortisol. And cortisol can negatively impact the body if you produce too much or too little of it. So we're going to talk about that towards the end of today's lecture. And I'm also going to discuss the epigenetics of stress and looking at how chronic stress can actually modify our gene expression. So originally it was assumed that the peripheral glands of the body, like the pancreas, the adrenal medulla, and the ovaries, simply knew what to do on their own in order to secrete hormones. However, new evidence has brought light to the secretory hypothalamus, and it turns out that the hypothalamus is kind of like the captain of the endocrine system, and it's responsible for commanding these other areas of the body to produce hormones. Now, the hypothalamus sits right here underneath the third ventricle and the thalamus, and ventral to this area is the pituitary. The pituitary gland has two different lobes we're going to talk about, and this posterior lobe right here is actually a part of the brain, whereas the anterior lobe shown here is an actual gland that sits right outside of it. And we're going to talk about that later on when we talk about hypothalamic control of the pituitary specifically. The hypothalamus is largely responsible for maintaining homeostasis in the body. And homeostasis is really just the maintenance of our body's internal environment. So thus, the hypothalamus can actually recognize different factors in our blood that can affect our health. So for example, the hypothalamus regulates blood volume, blood pressure, salinity, which is salt content in the blood, acidity levels in the blood oxygen levels, and blood glucose levels. This is just one of many mechanisms that the hypothalamus is responsible for. And interestingly, we're going to talk about how the hypothalamus can actually register how much salt we have in our blood, and it can actually tell the pituitary gland to produce a neurohormone called antidiuretic hormone, or vasopressin, which is dumped into the bloodstream when we're dehydrated and brings on those strong feelings of thirst. So ultimately, the hypothalamus is in charge of regulating many things in the blood. So as I mentioned, the hypothalamus is like this master gland of homeostasis, but it performs many other functions. It's involved in things like thirst and water intake, our hunger signals and satiety signals. It plays a role in the autonomic nervous system response. It helps us to regulate body temperature, blood pressure. It plays a role in our sex drive. So ultimately, We're going to talk about all of these different functions of the hypothalamus and the hormones that are produced due to its commands. There are 3 zones of the hypothalamus, including the lateral zone shown here, and the medial zone, and then the paraventricular zone, which is shown here in yellow. The paraventricular zone is particularly important to talk about. So the cells of the paraventricular zone lie right next to the wall of the third ventricle. And there are neurosecretory cells that live in the paraventricular zone and secrete hormones into the bloodstream. So we're going to largely be talking about these neurosecretory cells that live in the paraventricular zone of the hypothalamus. And it's important to note that this area of the hypothalamus does receive inputs from other zones of the hypothalamus, such as the lateral and medial zones, and it receives inputs from the telencephalon. So that would include some of those important forebrain structures we've been talking about in class, like the cerebrum, and it also receives input from the brainstem. It also regulates hormone secretion, as I mentioned earlier, but there's one group of cells in the paraventricular zone that I do want to talk about, and they are called the suprachiasmatic nucleus. This lies right above the optic chiasm. The suprachiasmatic nucleus is this important body of neurons, and their nuclei live here. These nuclei are responsible for synchronizing the circadian rhythms that we experience every day with the light-dark cycle. So these cells are actually receiving direct retinal innervation and function to synchronize these circadian rhythms. Therefore, it functions like the brain's master clock. So for example, when you wake up in the morning and sunlight is coming into your room, the suprachiasmatic nucleus is going to communicate with the pineal gland. And during this time, because it's light outside, it's going to command the pineal gland to secrete lower levels of melatonin. Whereas at night, during that dark cycle, the suprachiasmatic nucleus can recognize these cues and communicate with the pineal gland to produce higher levels of melatonin that will make us feel sleepy. Essentially, these cells are pretty incredible. Studies have shown that if you actually remove the suprachiasmatic nucleus, plate it in a petri dish, and allow them to grow, these cells can actually monitor circadian rhythms even outside of the body. So they truly are the brain's master clock. Now, the autonomic nervous system is largely controlled by the paraventricular zone of the hypothalamus. So remember, the autonomic nervous system is controlling our involuntary movements and things that are outside of our conscious control. So unlike the somatic nervous system, this is an unconscious system, but it's highly coordinated. The systems within the autonomic nervous system that we've already discussed include the sympathetic and the parasympathetic systems. But now we're going to talk about the enteric nervous system, which is also activated during the parasympathetic nervous response to aid in digestion. Along with the handout I created for y'all with the different nervous systems and how they come together, you can use this as a study tool to show the connection between the central nervous system and the peripheral nervous system. So this image is showing us the sympathetic and parasympathetic divisions of the autonomic nervous system. Now, I know this looks like a lot of information, so first we're just going to start by talking about the sympathetic division here in blue. So first, all of these neurons here that are labeled green, these long axons that are projecting to all of these different areas, are noradrenergic neurons. So remember that these are neurons that are largely producing and secreting norepinephrine. These neurons can synapse into areas like the heart, the liver, and the kidneys. And so norepinephrine is involved in our fight or flight response, but I want to note that the sympathetic nervous system isn't just involved in the fight or flight response. This is an easy way for us to understand how this system works, but the sympathetic nervous system response is largely activated during arousal and attention, so we require it to learn new material. It helps with us with cognition, and it even plays a role in our happiness and our mood. Additionally, the sympathetic system is responsible for stimulating glucose production and release. So essentially, norepinephrine can largely stimulate this glucose production and release. Additionally, norepinephrine is involved in the production and release of different hormones. There's a stress hormone or steroid called cortisol that is released from the adrenal medulla of the kidneys, and we're going to talk about how the hypothalamus is largely commanding this response during the stress response. Now over here, this is the parasympathetic division in green. And all of these neurons with these long projections here are cholinergic neurons. So they are secreting acetylcholine. So we know that these neurons can project and synapse onto different areas throughout the body. So different organs like the heart, the stomach, and the small intestines. For example, we've learned about the muscarinic acetylcholinergic receptor. Remember that these receptors sit on the cardiac muscle of the heart, and when acetylcholine is released here, they can aid in slowing down heart rate. So this is helping the body to rest. Also, these cholinergic neurons are involved in feeding behavior, physiological processes associated with the anticipation of food, and they're activated by hunger hormones. We're going to talk about how the hypothalamus largely commands these actions. The hypothalamus wants to keep the body in homeostasis and thus can command these satiety signals or these hunger signals in the form of hormones. And don't worry, we're going to talk about that later on. Additionally, the parasympathetic nervous system plays an important role in insulin and glucose homeostasis. So for example, after we consume a large meal, our body needs to produce insulin from the pancreas, shown right here. The pancreas is going to produce insulin, so that insulin, which is a neuropeptide by the way, can shuttle glucose where it needs to go for long-term storage. If you remember way back in the earlier lectures, we talked about how insulin is a neuropeptide, and neuropeptides are actually carried along axons differently than neurotransmitters. For example, they are held in secretory granules because of their larger size. And this particular neurohormone is going to be released from the pancreas during the digestive response. So during digestion, insulin is going to take that newly created glucose that was broken up by our food sources, our energy source, and it will take glucose to different areas of the body. For example, glucose can be stored in the form of glycogen in our muscles, and it can also be stored in the form of triglycerides in our white adipose tissue. And so a lot of this tissue, for example, visceral fat, sits around our organs. So thus, the parasympathetic system and the sympathetic system are largely modulated by commands of the hypothalamus, but during this parasympathetic response, the hypothalamus is helping us and helping insulin essentially store glucose for energy that will be required later on, whereas the sympathetic nervous system is going to be utilizing that glucose that was already stored during the parasympathetic nervous system response. Now we're going to discuss the diffuse modulatory neurotransmitter systems. We're going to talk about different types of neurons, such as cholinergic, serotonergic, dopaminergic, and noradrenergic neurons, and where their nuclei live throughout the brain. This is important because these neurons need to create a system, and the neurons in these systems might contact almost 100,000 other neurons. So they are working together in this synchronous fashion. First, let's talk about the acetylcholinergic system. Now, if you remember, in my earlier lectures, I talked about how this system is disrupted in the context of Alzheimer's disease. In Alzheimer's disease, there is a death of cholinergic neurons in the basal forebrain, and this can largely project back to the hippocampus and the medial temporal lobe. So thus, making sure that you get enough dietary choline is really important because dietary choline is how our body creates acetylcholine. And if you remember, some of those food sources include leafy greens, eggs, peanut butter, and Brazil nuts. Now, there are two key areas where acetylcholinergic neurons live. This includes the medial septal nuclei, shown here, and the basal nucleus of Maynard. These areas are where acetylcholinergic cells largely populate, but they have Golgi type 1 axons. So these axons are going to be able to project to other regions throughout the brain. So this diffuse modulatory system can play a role in our learning and our memory because it does project to the hippocampus, and it also projects to the thalamus before it can go to the cortex. This system can also project down the brainstem to the through the pons in the medulla, which has important functions for regulating our sleep and our blood pressure. It's actually involved in turning REM sleep on and off. It plays an important role in attention and alertness. So thus, we've talked about how acetylcholine is released during that parasympathetic nervous system response. And it's also really important for cognitive function. So as I mentioned previously, the death of cholinergic neurons is one of the beginning stages of Alzheimer's disease pathologies. Now, the serotonergic system is largely diffusing from the Raphe nuclei. So the Raphe nuclei shown here are these large red dots, and this is the cell body or somas of the serotonergic neurons that live within the brainstem. And again, they have long axons, Golgi type 1 axons, that can project into all of these different areas of the brain. So the Raphe nuclei release serotonin, which then can diffuse throughout the brain. Of course, we know that serotonin plays an important role in mood and depression, as we've talked about the treatment of generalized anxiety disorder and depression with selective serotonin reuptake inhibitors, which prevent the reuptake of serotonin in the synaptic cleft, in addition to monoamine oxidase inhibitors that prevent that enzymatic breakdown of monoamines like serotonin. Additionally, serotonin actually plays a role in our feeding behavior, which I always laugh because feeding and eating makes me happy. So no wonder it's involved in the serotonergic system. But serotonin is also released during that process. Also, serotonin is involved in pain and pain processing, and even in our wakefulness and our sleep cycle. And here's the dopaminergic system. So we have pockets of dopaminergic cells in the ventral tegmental area and the substantia nigra, which we've already largely talked about before. So these two areas are where the nuclei or the actual cell bodies of the dopaminergic neurons live, and their axons can project to other areas of the brain. So the cells in the ventral tegmental area actually extend through the limbic system into the frontal lobe, and then cells in the substantia nigra can extend to the striatum. Now, if you remember, the striatum is made up of the caudate nucleus and the putamen, and these structures together make up the basal ganglia. So thus, the basal ganglia, of course, is largely allowing us to plan out our motor movements and coordination. And so the basal ganglia is heavily involved in movement, but these areas, of course, are also important for our reward and pleasure systems. So the projections here actually stop at the nucleus accumbens, which is right under the caudate. And so this is a reward center of the brain. So anytime we do something that's pleasurable, that feels good to us, this area is going to be activated. The ventral tegmental area can largely be hijacked by drug addiction or alcoholism because this is a reward area of the brain as well. And so this system is going to be activated due to addictive behaviors. Thus, it also plays an important role in motivation, and it also plays an important role in cognition, which we're going to talk about later on. And here's the noradrenergic system. So this is a system of neurons that are producing the neurotransmitter norepinephrine. And so the cell bodies of these neurons are in the locus coeruleus. This actually means blue spot or blue place. And these are clusters of brain cells that have many synapses. And their axons can project to other areas. These are Golgi type 1 axons. They can project all the way to these other areas of the brain in the cortex, to the cerebellum, and even into the spinal cord. So norepinephrine is largely involved in alertness and awakeness. So this is really important because they are involved in the sympathetic nervous system response. Norepinephrine is also involved in anxiety and depression and mood changes. There are actually certain drugs that psychiatrists can prescribe for patients that are undergoing anxiety and stress that are going to help with depression symptoms and help to increase the amount of norepinephrine that is available. And norepinephrine also plays an important role in learning and memory. So here's an overview of everything we've talked about so far concerning the nervous system and the different connections that make up the nervous system. First, I introduced the central nervous system, which is made up of the brain, the brainstem, and the spinal cord. We've talked about efferent motor neurons that emerge and exit the brain that command muscle movement, and they are going to innervate the spinal cord at the ventral root. We've also talked about afferent sensory neurons that are going to be receiving sensations and sending them up to the central nervous system. So thus, afferent sensory neurons are moving away from the body to allow the brain to process different sensations that we feel. Now we've talked about the peripheral nervous system and the two subsystems, including the somatic and the autonomic nervous system responses. Now remember, the somatic nervous system is largely in charge of our voluntary movement. So these nerve cells are going to innervate our skin, our joints, and our skeletal muscle. And we have efferent motor neurons that are exiting the brain that can actually command these muscle movements. And then, of course, the autonomic nervous system is controlling involuntary movements outside of our conscious control. So these nerves are largely innervating our internal organs and smooth muscle, blood vessel, and glands. So, for example, it innervates the small intestines and the heart. And these systems, broken down into further systems, are called sympathetic and parasympathetic systems. We've talked about the sympathetic system now in more depth, and how it's not just involved in fight or flight response, but in arousal and alertness. We've talked about the parasympathetic system in more detail and how it's helping the body to regulate homeostasis. It's involved in digestion and preparing the body with energy storage for future use. Now, efferent motor neurons are going to be commanding movements for both of these systems, whereas afferent sensory neurons from these systems can synapse back up to the central nervous system, so the brain is made aware of these sensations in the periphery of the body. The last system I haven't talked about in depth is the enteric nervous system. This is the mini brain in our gut, and we're going to talk about how the gut actually plays a really important role in our mood, our behavior, and our cognition. So the enteric nervous system, as I mentioned, is basically like a mini brain in our gut. There are over 500 million neurons, equal to the number of neurons in the spinal cord that live in the gut, for example, in the small intestine shown here. Now, this area in the enteric nervous system is largely inhibited during strong sympathetic nervous system activation. So, for example, during the sympathetic nervous system response, this is not a good time for us to worry about digestion and shuttling glucose for long-term storage, right? The sympathetic nervous system response is going to need to utilize that glucose. It needs to stay alert during these processes. So thus, this little brain in our gut is largely inhibited during that response. But the enteric nervous system plays an important role during the activation of the parasympathetic nervous system response, during that rest or digest response. So I'm just briefly going to talk about the enteric nervous system, but essentially it communicates with the brain via nerve cells in the peripheral nervous system. So remember, nerve cells in the peripheral nervous system are referred to as ganglia. So the enteric nervous system exists in the small intestine and the colon, and there are all of these interneurons here. So remember, these are neurons that synapse onto other neurons and other types of neurons that are playing a role in reflex pathways. They're helping to control patterns of contraction for bowel movements, helping with blood flow and movement of fluids. So the central nervous system is in control of the esophagus, the stomach, and colorectal functions, but it's working together with the enteric nervous system in the gastrointestinal tract specifically. Now, here are some of the pathways from the central nervous system and how they can reach the enteric nervous system down here. So there are different pathways such as vagal pathways, sympathetic, and pelvic pathways. Now, I really wanted to primarily focus on these vagal pathways. Now, we briefly talked about the vagus nerve before. This is known as the wandering nerve. The vagus nerve is a cranial nerve that is a part of the peripheral nervous system that innervates several different organs in the body. It's known as the wandering nerve because it innervates many areas of the body, such as the stomach, such as the intestines, and it's helping direct communication between the enteric nervous system and the central nervous system. And I did want to note that there is a special population of cells here in the neck called the nodose ganglion. The nodose ganglion can essentially help with communication between the central nervous system and the enteric nervous system via transduction of the vagus nerve. So as I mentioned previously, we have over 500 to 600 million neurons that are in the enteric nervous system. And recently, there was a research group at Duke University that discovered these nerve cells in the enteric nervous system called neuropod cells. So if we look at this image here, this is a section of mouse intestine. So remember, to get these images, you would have to use something like a microtome where you can take extremely thin sections of tissue, which they are put on a slide and fixed to a slide, and scientists can stain to look at different things. So here in this image, the reason that I have neuropod cells highlighted in green is because the cells here that are green are neuropod cells themselves. So there are very few neuropod cells in this picture, but this finding was really amazing, especially for the science of the gut-brain axis, because it showed us that these nerve cells in the enteric nervous system are playing a role between communication with the gut and the brain. So neuropod cells, in addition to the nodose ganglia up in the neck, are helping to form this connection, and usually they are communicating via transduction of the vagus nerve. Interestingly, neurons in the enteric nervous system can actually produce and secrete neurotransmitters. 90% of the serotonin that we create is actually created in the gut. That doesn't necessarily mean that all of that serotonin is going to get up to the brain, though. I do want to note that serotonin is different from brain-derived serotonin that's created in the brain. But thus, these neurotransmitters in the gut can help to regulate our mood, as we know that the gut and brain are connected via transduction of the vagus nerve. So this is important to note, that having a happy gut can actually help improve your mood. Studies have shown that if you have decreased microbial species in the gut, this is associated with increased risk of anxiety and depression. So for example, we have a very large network of gut microbes in our body. These gut microbes together form our microbiome. So we actually have more microbes in our body than we do our own cells. And scientists have estimated that if you removed our gut microbes, they would be over 6 pounds. So it's pretty incredible to think that we're just largely this petri dish of bacteria. And we need to take good care of our gut microbes so they can take care of us. That's why it's so important to consume prebiotic fiber, because when we digest fiber, those microbes are actually the ones that are doing the work to break down that fiber to produce metabolites such as short chain fatty acids. A great example of a short chain fatty acid is butyrate. Butyrate is going to help with cognition. Studies have found that butyrate supplementation can actually improve anxiety-like behavior and learning and memory in rodents. And also, these gut microbes need to have diverse populations. And the way that you can nurture your gut microbiome is by consuming probiotics. This is going to ensure that you have a proper ratio of these gut microbes, meaning you're going to have a good balance of those essential commensal gut bacteria that are going to help break down fiber. And you also have a balance with those maybe gram-negative bacteria, the bad bacteria in your gut. A way that you can help those good commensal bacteria flourish is through probiotics. Again, you can take that in supplement form, but scientists have actually found that it's much more helpful to get probiotics through dietary sources like kombucha, kefir, kimchi, and yogurt. So these fermented foods. You can also get this in sauerkraut as well. If you're ever interested in hearing more about probiotics and prebiotics, please reach out to me because this is something that I also teach about in my nutritional neuroscience class. So now we're going to talk about how the hypothalamus is in control of the pituitary gland and how that affects our endocrine system. So here is the pituitary gland. It sits ventrally to the hypothalamus. And there are two lobes of the pituitary, the posterior lobe shown here and the anterior lobe here in the front. So the hypothalamus controls these two lobes in different ways. And I do want to note that the posterior lobe of the pituitary is actually part of the brain, whereas this anterior lobe is actually a gland that's responsible for secreting hormones. And the pituitary is gently held in a cradle of bone at the base of the skull. So now let's talk about the hypothalamus and its control over the posterior pituitary. So again, this is the part of the pituitary that's actually a part of the brain. As I mentioned at the beginning of this lecture, the hypothalamus has these important cells that are called neurosecretory cells. Neurosecretory cells are largely commanding the secretion of hormones into the bloodstream. And so the posterior lobe of the pituitary gland is receiving commands from these specialized neurosecretory cells in the paraventricular nucleus. And so these cells are called magnocellular neurosecretory cells. So their cell bodies live in the paraventricular zone of the hypothalamus, but their axons extend down to the posterior pituitary gland. So the posterior pituitary is responsible for producing and releasing neurohormones. So these are substances that are dumped into the bloodstream that can help us with many different factors, but there are two that I'm specifically going to talk about, and this includes oxytocin, which is our bonding hormone, and vasopressin. Vasopressin is also known as antidiuretic hormone, and it plays an important role in thirst. As I've mentioned previously, oxytocin is our love neurohormone or our bonding hormone. It's released when we're engaging in intimate behaviors, when we're cuddling with our partner. It's also released when a mother or father is bonding with their newborn child. So essentially, this hormone allows us to form connections with our loved ones. It also plays a really critical role during the final stages of childbirth. So it can cause the uterus to contract and facilitates the delivery of the newborn. And it also stimulates the ejection of milk during breastfeeding. So, interestingly, all lactating mothers know about this complex letdown reflex that involves oxytocin neurons of the hypothalamus. Oxytocin release can actually be stimulated by the somatic sensations generated by a suckling baby or even the sound or sight of a baby. So, for example, when the brain receives a sensory stimulus, this could be somatic, visual, or auditory, it can reach the cerebral cortex via the thalamus. We know that the thalamus is the brain's relay station. So, interestingly, any type of visual or auditory cue, like the sound of a crying baby, can trigger the release of milk beyond a mother's conscious control. So even if someone else's baby is crying and it's not your newborn baby, the hypothalamus can trigger oxytocin release to stimulate milk release. So ultimately, this obviously makes sense. This is very advantageous for women to be able to start secreting breast milk when their baby is crying, because this is usually a sign that they need to feed their baby. So the hypothalamus is playing a really important role in this letdown of milk. Also, another neurohormone that is produced due to these magnocellular neurosecretory cells in the paraventricular zone of the hypothalamus is vasopressin, which again is synonymous with antidiuretic hormone or ADH, and this helps to regulate blood volume and salt concentration. So when our body is deprived of water or we are dehydrated, our blood volume actually drops and our blood salt concentration increases due to the smaller volume of blood. These changes can be detected by receptors within the cardiovascular system, and the hypothalamus specifically can register salt concentrations in the blood. So these vasopressin containing neurons receive information about these changes and respond by releasing vasopressin. So we're going to talk about this system and how the subfornical organ, hypothalamus, kidney, and liver are all related in helping us to produce this hormone that's going to cause us to drink water. So vasopressin acts directly on the kidneys, and it leads to water retention and reduced urine production. So let's go through this image step by step. So first, if we have conditions of lower blood volume or pressure, this means that we have higher salt content in the blood. And this is usually when we're dehydrated and we need to drink water. So during this time, the kidneys are going to be secreting renin, this protein, into the bloodstream. And this actually promotes the synthesis of this peptide here known as angiotensin 2. Angiotensin 2 is going to be released into the bloodstream and reach the subfornical organ. That sits right here dorsally to the hypothalamus. The subfornical neurons are going to stimulate the hypothalamus because they have axons that can communicate with the hypothalamus. And this is going to increase vasopressin release, or ADH, from the posterior pituitary. This is going to induce the production and feeling of thirst. Now let's talk about how the hypothalamus can control hormone release from the anterior pituitary. So here's the anterior lobe of the pituitary, which remember is an actual gland outside of the brain itself. And so there are special neurosecretory cells that live in the hypothalamus that again can travel down and have axons that synapse into the anterior lobe of the pituitary. The cells in this lobe can synthesize and secrete a wide range of hormones, and they can also regulate the secretion of hormones from other glands in the body, such as the gonads. So together, this entire system, including the hypothalamus, where those neurosecretory cells are commanding the anterior pituitary lobe, and then The anterior pituitary's hormones that are going to also be regulating hormones released from other glands in the body completely create our endocrine system. And so, again, as I mentioned, the pituitary hormones can act on other glands like the gonads. They also can affect the thyroid glands, the adrenal glands, and the mammary glands. So, what controls the anterior pituitary? You guessed it, the hypothalamus. The hypothalamus is truly the master gland and essentially the captain of the endocrine system. So, it's going to be commanding the secretion and production of these hormones that are going to be dumped into the bloodstream. Now, let's talk about the hormones of the anterior pituitary specifically. There are several different hormones that are released by the anterior pituitary. When we look at this image, we're just going to focus on the anterior pituitary. So this is shown here in blue. Over here, this is demonstrating different neurohormones that are released from the posterior pituitary, which we already talked about. So the anterior pituitary is largely going to be releasing hormones that can target other glands like the gonads, the thyroid, and the adrenal cortex on the kidney. So, for example, we have growth hormones, so this is known as GH, that can be released from the anterior pituitary and can actually stimulate protein synthesis in our bones and other areas of the body. Also, the anterior pituitary is responsible for the release of thyroid stimulating hormone, or TSH. This can play a role in thyroxine secretion by the thyroid. Additionally, we're going to talk about how the anterior pituitary releases this hormone called adrenocorticotropic hormone, or ACTH. And so this hormone is released into the bloodstream by the anterior pituitary, and then it's going to reach the adrenal medulla of the kidneys. Once it reaches this area, the kidney is going to respond to ACTH by creating a stress steroid known as cortisol. Cortisol helps us to mobilize our energy stores during the fight or flight response, or when our body is stressed. It's going to require energy. So cortisol is doing that. And we're going to talk about how cortisol is released throughout the day, and it's largely released in the morning when we first wake up. Finally, the anterior pituitary releases other important sex hormones like follicle stimulating hormone, which is going to target the ovaries and the gonads. It's helping with ovulation and the production of sperm. It also produces luteinizing hormone, or LH, which helps with ovarian and sperm maturation. So ultimately, the anterior pituitary, although it's a very small gland, has a lot of different roles in inducing hormone secretion that's going to affect these other areas and glands and organs of the body specifically. So the anterior lobe of the pituitary gland is under the control of neurosecretory cells that sit in the paraventricular zone of the hypothalamus. So remember, this is similar to the neurosecretory cells that are commanding movement of neurohormones out of the posterior pituitary. But if you remember, those cells were called magnocellular neurosecretory cells. The neurosecretory cells that are commanding hormone release in the anterior pituitary by the hypothalamus are called parvocellular neurosecretory cells. And they communicate with their targets via the bloodstream. So again, these parvocellular neurosecretory neurons that live in the paraventricular zone of the hypothalamus secrete something called hypophysiotropic hormones. These are secreted at the floor of the third ventricle. And eventually they make their way down to the anterior pituitary lobe, where they're going to bind to receptors on the pituitary cells. This is going to activate these receptors in the anterior pituitary to either secrete or inhibit the release of hormones into the bloodstream. So, how is the hypothalamus involved in the stress response? We know that cortisol, a steroid, is largely modulating the stress response. So, cortisol is a steroid and it's a class of biochemicals related to cholesterol. And so, cortisol is released into the bloodstream to help us mobilize our energy reserves. So, to take up that glucose that's stored in the form of glycogen or triglycerides in the white adipose fat. It can also suppress the immune response because, of course, if your body sympathetic nervous system is activated, that fight or flight response is more important at the current time than maintaining the body's immune system. So, from an evolutionary perspective, this is advantageous because, for example, if your body is undergoing an acute physiological stressor, you're going to require energy reserves and you're not going to want to dedicate that energy to increasing the immune response. However, we're going to talk about how long-term stress, usually induced by both physiological and psychological stress, can unfortunately suppress the immune system due to increased cortisol production. And that kind of stress and cortisol release can actually lead to some really negative health effects. And I'm going to talk about that in a few slides. But I do want to note that cortisol is important. So it's not a bad thing. We need cortisol release to help us with our stress. Cortisol is a positive thing in helping with physiological stress, positive emotional stimulation, and psychological stress. So acute release of cortisol and the normal basal amounts of cortisol that the body secretes is beneficial to us. It's when we either create too little or too much cortisol that can affect our health and our largely long health consequences. As I noted previously, the parvocellular neurosecretory cells in the paraventricular zone of the hypothalamus are connected to the cells in the anterior pituitary gland. So the hypothalamus is commanding the release of different hormones from the anterior pituitary. The anterior pituitary is also connected to the adrenal cortex on the kidneys. As shown right here, this is the adrenal medulla and adrenal cortex on top of the kidney. And so essentially, these cells in the hypothalamus are activated during the stress response. And if we are experiencing a stressful situation, the body is going to respond by producing this stress hormone called cortisol. Now, these neurons release a peptide that's known as corticotropin releasing hormone, or CRH, into the blood. So this is when our sympathetic nervous system is activated, our hypothalamus is going to, the parvocellular neurosecretory cells in the hypothalamus specifically are going to induce this release of corticotropin releasing hormone into the blood. CRH is going to travel a short distance because it's just going to go right down here to the anterior pituitary. And the anterior pituitary is going to stimulate the release of a hormone known as adrenocorticotropin hormone, or ACTH, as shown here. When ACTH is dumped into the bloodstream from the anterior pituitary, it's going to reach the kidney. When it reaches the kidney, it is going to travel to the adrenal cortex of the kidney specifically, and ACTH is going to induce the release of the steroid cortisol. This is what's known as the HPA axis, which stands for hypothalamic anterior pituitary adrenal medulla axis. Now, the HPA axis is going to be activated when the body is undergoing stress. Unfortunately, our ancestors were usually undergoing physiological stressors, but now in today's society, we are undergoing both physiological and psychological stress. So thus, these stressors are constantly activating our HPA axis. And while acute activation of the HPA axis can be very beneficial in helping us deal with stress, long-term activation of the HPA axis can render serious health consequences. And later on, we're going to talk about the effects of stress on the body. So what happens if you don't produce enough of the steroid cortisol? This is known as adrenal insufficiency, and there's a rare disorder called Addison's disease in which a person can experience degeneration of the adrenal gland on the kidney, and thus they do not produce enough cortisol. So, interestingly, John F. Kennedy actually had Addison's disease, and this was largely concealed during his presidency to protect his youth and his vigorous image. But Kennedy required a daily hormone replacement therapy to compensate for the loss of cortisol. So, unfortunately, a loss of cortisol can really affect the body because we know that cortisol plays a really important role in our attention and how we modulate the stress response. So, unfortunately, due to this lack of cortisol, he experienced extreme fatigue, weight loss, and poor appetite from reduced blood pressure and blood sugar. So, Addison's disease is treatable, it is rare, but it can be treated with cortisol replacement therapy. What happens if you produce too much cortisol? So, the flip side of adrenal insufficiency, also known as Addison's disease, is a condition called Cushing's disease. This is caused by pituitary gland dysfunction that results in elevated levels of ACTH. So, thus, if you remember, ACTH is released into the bloodstream by the anterior pituitary. And when it reaches the adrenal medulla on the kidneys, it's going to help us produce the steroid cortisol. However, elevated levels of ACTH are going to further increase our levels of cortisol, which can actually render some serious consequences like rapid weight gain, immune suppression, sleeplessness, memory impairment, and irritability. So, as I mentioned previously, high levels of cortisol can suppress the immune system because the sympathetic nervous system is activated and the body is responding to stress. Long-term immune suppression can be really unhealthy. Obviously, the immune system needs to be active to help protect the body against pathogens. And of course, cortisol plays a role in alertness, so it can affect Cushing's patients and induce insomnia or sleeplessness. And of course, this is going to make these patients irritable. So Cushing's disease is different from Addison's because it leads to a higher spike of cortisol, and it's going to render opposite effects of Addison's disease. Now I'm just going to quickly summarize the pituitary. So here's our big picture. This is summarizing the pituitary gland, so I'm going to quickly recap. The posterior pituitary is a part of the brain. It's connected to the hypothalamus because if you remember, there are magnocellular neurosecretory cells that live in the paraventricular zone of the hypothalamus, and they have long axons that project into the posterior pituitary. The hypothalamus serves as a captain, essentially, helping to release neurohormones such as oxytocin, our bonding hormone, a hormone that also plays a role in contractions during childbirth and during breastfeeding for stimulating milk release. And then another neurohormone we talked about was antidiuretic hormone, ADH, which is also known as vasopressin. And vasopressin is released when we are feeling thirsty due to lower blood volume and higher blood salinity. Second, we talked about the anterior lobe of the pituitary, which is actually a gland that sits outside of the brain. And we talked about parvocellular neurosecretory cells that communicate with cells in the anterior pituitary. And this induces the release of several different hormones throughout the body. These hormones that are secreted and dumped into the bloodstream by the anterior pituitary include thyroid stimulating hormone, adrenocorticotropin, growth hormone, and different hormones that target the gonads, such as follicle stimulating hormone and luteinizing hormone. Now, I mentioned earlier how I was going to talk about the evolution of stress and how our ancestors were largely undergoing physiological stressors, whereas in more modern times, we are dealing with chronic psychological stressors that are affecting our health. So here's Hans Sali. He's kind of known as the godfather of stress physiology, and he performed many different experiments to try to understand how stress blunts the immune response and how that affected animal behavior specifically. In one of his experiments, this was really odd, but he injected ovarian fluids into rats. And this obviously induced different hormone changes in the rats, which caused enlarged adrenal glands. And enlarged adrenal glands, remember that is on the kidney, increased the amount of cortisol that was being produced. This led to a shrunken immune tissue, so different immune organs, and prolonged stress blunted their immune response. These rats also developed peptic ulcers. So they were literally so stressed they got ulcers. I know we hear that joke all the time, like, oh, I'm so stressed I could get an ulcer. But in fact, the stress response did create ulcers in these animals. Now, stress is this process in which a stimulus is evaluated. We can see it as threatening or potentially insurmountable. This can elicit a really deep disturbance of our mental health, and it can lead to different behavioral and biological adjustments so that the body can try to regain homeostasis. Now, previously, as I mentioned, we encounter physiological stressors, but for the most part today, our stressors are psychological. These deeply rooted stressors that can make us anxious. So sometimes we sadly have this chronic worry. Now, there's a famous scientist at Stanford University. His name is Dr. Robert Sapolsky, and he has been studying stress for most of his career. He wrote this book here called Why Zebras Don't Get Ulcers. I definitely recommend it. It's a great read, and it essentially talks about how we need to try to catch up and adjust to chronic worry. Unfortunately, we are undergoing this chronic psychological stress that our body has not really figured out how to adapt to. So thus, because we're constantly undergoing stress, our sympathetic nervous system is constantly activated. And from an evolutionary standpoint, we were not meant to live with these overactivated sympathetic nervous systems. So we are so prone to the negative effects of stress because of our psychological stress. The whole point of Dr. Sapolsky's book, Why Zebras Don't Get Ulcers, has a really interesting thesis. So our stress response is very well attuned to physical stressors. So for example, for a zebra, a physical stressor would be running away from a predator like a lion. Now, a zebra is going to have an activated sympathetic nervous system response when it sees a lion. It's going to go into fight or flight mode. And we know during this time, the stress steroid, cortisol, is going to be released. The zebra is going to require cortisol to help collect glucose from stored tissues, and it's going to utilize that glucose to increase heart rate, to increase blood flow, and run away from that stressful, scary situation. However, once a zebra gets away from a predator, it can go right back to homeostasis. So these are acute physical stressors that aren't going to be happening for long periods of time. Whereas our current physiology in humans hasn't caught up with our new types of stressors. These ongoing psychological stressors that can keep us up late at night that we're constantly worried about. Maybe we're worried about our relationships, finances. Maybe we're stressed about studying for an exam. These are all going to be in our thoughts, and it's not an acute stressor that we can just shake off like running away from a predator. So interestingly, Ulcers are actually formed from a bacterium called Heliobacter pylori. This is known as H. pylori, and this lives in our stomach. It's involved in the creation of ulcers, which are in the stomach lining specifically. So again, as I mentioned, H. pylori is a bacterium. It's found in the gut, but it's also been discovered in pond water. I know that sounds kind of gross, but if this bacterium is present in your gut microbiome and you're undergoing severe chronic stress at the same time, you actually can develop ulcers. But you can't develop an ulcer without H. pylori present in your stomach. So chronic stress has actually been shown to induce thinning of your stomach lining, and this can further increase your likelihood of developing an ulcer. So ultimately, chronic stress has a lot of terrible long-term effects on the body. And these can be both physiological and psychological. So there are several risk factors for Alzheimer's disease. We know that there are different environmental risk factors specifically that contribute to the generation of sporadic Alzheimer's disease, which is also known as late onset Alzheimer's. This is a type of Alzheimer's disease that's usually discovered after the age of 65. And although we don't know the actual root cause of this disease, we know there are several environmental risk factors that contribute to its development and progression. And one of them is chronic stress. Sadly, about 77% of the US population reports that they experience stress, and this stress affects both their mental and physical health. Individuals that have MCI, which stands for mild cognitive impairment, who also undergo chronic stress have an increased risk of developing dementia. MCI is often one of the very first symptoms of dementia and Alzheimer's disease, and a clinical test can be actually utilized to see if an older individual has signs of mild cognitive impairment. Elderly individuals who are more prone to stress were more likely to develop Alzheimer's in a previous human subject study. So ultimately, long-term stress isn't healthy for the body, and it can increase risk of neurodegenerative diseases such as Alzheimer's disease. Now let's quickly recap some of the main topics that we talked about throughout Chapter 15. First, we learned about the hypothalamus and how it's largely in control of the endocrine system. It controls the ventral pituitary gland below it in a few different ways. What are two lobes of the pituitary? First, we learned about the posterior and anterior pituitary. If you remember, there are two key neurohormones that are produced by the posterior pituitary, including vasopressin, also known as antidiuretic hormone or ADH, and oxytocin. We also learned about the anterior pituitary and how there are several different hormones that are secreted by the anterior lobe that are going to influence other peripheral organs, such as thyroid stimulating hormone or TSH that's released from the anterior lobe. And then this goes to the thyroid. We also learned about different sex hormones, such as follicle stimulating hormones that are released from the anterior lobe of the pituitary. These are dumped into the bloodstream that go down to the gonads and play a role in sperm production and ovulation. So ultimately, there are several different hormones at play in the anterior pituitary, and the hypothalamus is commanding these neurohormones and other hormones. We also talked about the HPA axis, known as the hypothalamic anterior pituitary adrenal cortex axis, and how this system is activated during the stress response. Now we're going to talk about stress and how chronic stress can actually influence our gene expression. So first, to understand gene expression and the way that stress can affect gene expression, I need to introduce something called epigenetics. This is the study of the modification in gene expression without actually alternating the genetic code. So essentially, these changes aren't actually changing the nucleic acid sequence, but it is changing the way that these genes are expressed. Now, remember, we know that the final product of gene expression is essentially protein synthesis. There are two main methods of altering gene expression, and the first is DNA methylation. This is replacing a hydrogen atom with a methyl group. In more simpler terms, this is just adding a methyl group. Adding a methyl group is going to affect transcription factors because they can't bind to DNA in these areas, and thus it's going to repress gene expression. Now remember, if gene expression is repressed, that means that proteins may not be synthesized from this particular gene. Now, histone acetylation is another form of altering gene expression, but this typically increases gene expression. So, for example, if you remember histones, that's the structure that DNA is wrapped around, plays an important role in packing up that long strand of nucleic acids. Adding an acetyl group here to the tail of a histone is going to actually allow it to open up. And it makes that part of the gene that was previously unreadable now readable. Transcription factors can come and bind here and increase the ability for these genes to be expressed. So in simple terms, for this slide specifically for your exam, I just want you to understand that DNA methylation and histone acetylation are both involved in altering gene expression, whereas the addition of these methyl groups usually represses the expression of genes, and histone acetylation, the addition of these acetyl groups will actually increase the ability for genes to be expressed. So DNA methylation is largely characterized by a gene known as DNMT, DNA methyltransferase. This regulates the addition of methyl groups to the nucleotide cytosine. Now, glucocorticoids are super important during the stress response. Glucocorticoids are these important stress hormones, and they can signal via these receptors called glucocorticoid receptors, shown here in turquoise. Now, glucocorticoids stimulate the production of glucose, and thus that's really important because we know that glucose needs to be shuttled from those long-term storage centers like glycogen in muscle or triglycerides in white adipose tissue that need to enter the bloodstream to provide energy for the fight or flight response. Now, when our bodies actually respond to stress, we activate or inhibit the actions of glucocorticoids, and this is a protective mechanism. Glucocorticoid receptors are necessary to protect our bodies from the adverse effects of chronic stress. We need plentiful glucocorticoid receptors where glucocorticoids can actually come and bind to them. This is going to help modulate the stress response, and this is a very good and adaptive mechanism. Interestingly, the effects of stress in maternal care have been studied by many scientists, and Michael Meany is a very important researcher who looked at the effects of maternal care on epigenetics in offspring. So we're going to talk about his experiments in much more depth. So first, you're probably laughing looking at this, wondering what low licking and grooming means. So some rat moms will lick their pups a lot, and this is a sign of maternal care. So for example, just like a human mother would take care of their newborn, spend time with their newborn, or clean their newborn, this goes for the same for rat moms. They need to care for their offspring. Studies have found that if rat moms spend more time licking and grooming their pups, this can help the pups during their development to have a healthier health outcomes. So low licking and grooming has been associated with these more negative effects in offspring. So here's an example of that in Michael Meany's study. He found that pups that received lower levels of maternal care, meaning their moms did not lick them as often, they had decreased glucocorticoid receptors. Now, as I mentioned previously, glucocorticoid receptors are actually really important for helping us modulate the stress response. If we have more glucocorticoid receptors present, we are better able to adapt to stress. These pups also had higher levels of corticosterone. This is a form of cortisol in rodents. Additionally, once the pups were weaned from their moms, they found that when presented in different tasks that are able to measure anxiety-like behavior in rodents, such as the elevated zero maze, these pups presented more anxiety-like behavior and depressive behavior. Additionally, in the group of moms that were Hylic moms, meaning these moms were providing more maternal care to their pups and more bonding with their pups, there were better health outcomes associated with this. These pups had lower corticosterone levels, so remember that is a type of cortisol in rats. They had lower anxiety-like behavior, and they had increased glucocorticoid receptor expression. So ultimately, maternal care can actually affect the way that genes are expressed. So this is epigenetics at play. Here in image A, in the Lolic moms, we can see that methyl groups were added. This methylated promoter region of the glucocorticoid receptor gene decreased the expression of glucocorticoids. So these Lolic moms had diminished DNA demethylation and diminished histone acetylation for glucocorticoids. Whereas these moms that were providing more maternal care to their pups, this actually did induce upregulation or increase of glucocorticoid gene expression, which ultimately helped to protect the pups later in life. You could wonder if this occurs in humans as well. And we do know that better maternal care and even early diet, so the diet that is exposed to offspring throughout gestation and lactation, can induce better health effects later on by inducing these epigenetic changes, so increased expression of genes that could be helpful later in life in the prevention of different diseases. So ultimately, it's not just chronic stress that can actually induce these epigenetic alterations of DNA. So we know now that there are several different environmental factors, usually pro-inflammatory triggers that can alter the way that genes are expressed. Gene expression can change due to several different environmental factors, including our typical American diet that's known as the Western diet, obesity, chronic stress, infections, and allergies. But let's focus on the Western diet for a moment. Our typical American diet is characterized by being a high fat diet that is particularly high in saturated fatty acids. Saturated fat is commonly found in high fat dairy products, meat, and fried foods. Unfortunately, a lot of our fried foods today are fried in refined oils that are really high in either saturated fat or omega-6. Omega-6 is another type of fat that usually can be anti-inflammatory in the right dose, but high levels of omega-6 can become pro-inflammatory. The Western diet also includes a lot of high glycemic load carbs. So these are carbs that, when digested, cause a rapid increase in blood glucose levels. So obviously, this is going to increase blood sugar and increase risk of diabetes mellitus type 2. There are several different components of the Western diet that are contributing to this pro-inflammatory response, but studies have found that exposure to the Western diet throughout gestation and lactation can induce epigenetic alterations in rodent offspring. And we'll talk about that in a moment. But ultimately, all of these different factors can contribute to increased pro-inflammatory cytokine production. And if you remember, we talked about pro-inflammatory cytokines at the beginning of this course and how these are effector proteins that are released from immune cells during the innate immune response. Initially, the release of pro-inflammatory cytokines is advantageous to the body. Cytokines are usually released at areas of infection, and inflammation is usually increased by the body to help protect it in these damaged areas. But chronic production of pro-inflammatory cytokines can render serious consequences. So, for example, persistent inflammation can lead to chronic inflammatory diseases. There are several different chronic inflammatory diseases and autoimmune diseases like rheumatoid arthritis, cardiovascular disease, diabetes mellitus type 2, PCOS, and even dementia and Alzheimer's disease are characterized as inflammatory diseases. Now, as I mentioned previously, perinatal exposure to the Western diet has been shown to actually increase anxiety-like behavior in rodents. So in these experiments that I cited here, a dam, which is a rat mom, is exposed to a Western-style diet. So this would be a diet characterized with high saturated fat, high sugar levels, high glycemic load carbohydrates. The rat is exposed to the Western diet throughout pregnancy, so throughout gestation and lactation. When rat offspring are weaned from their moms and placed on a regular standard control diet that most rodents consume in laboratory settings, they're tested in different paradigms that can help us measure anxiety-like behavior. This paradigm right here is called the elevated zero maze. And it allows us to measure anxiety-like behavior because mice typically don't like these wide open spaces. They prefer to hide in these dark enclosed walls. So if the mice or rats spend more time in these open quadrants, this indicates that they have reduced anxiety-like behavior, whereas if they spend more time within the walls, this is demonstrating that they have increased anxiety-like behavior. And ultimately, in all of these studies that I cited, these researchers found that rodents that were exposed to a Western diet throughout gestation and lactation had higher levels of anxiety-like behavior compared to rodents who were just kept on a standard low-fat rodent diet. So ultimately, these changes from perinatal exposure to the Western diet need to be further explored to see how they can induce further epigenetic changes or actual alterations of the way that genes are expressed specifically.